Hello and welcome to this video where I'll be talking about multiple sequence alignment and in particular how it is used to represent patterns and profiles and how multiple sequence alignments can be represented in different ways. So we have kind of three objects that we'll discuss. The first are called patterns. And this includes things like consensus sequences. And then we have profiles or logos. And these are ways to look at the frequencies of amino acids or bases occurring in multiple sequence alignments in different ways. And in a later video, I'll be discussing how multiple sequence alignments are obtained. So here's an example of a multiple sequence alignment from an experiment. And what we're looking at here is an engineered phage genome or a segment of it. And here you can see an EM picture of a phage. This is not taken by me, but this multiple sequence alignment here shows some interesting things. And the first feature that kind of stands out, you'll see, is this region here, which unlike the other columns in this alignment, looks very random. And in fact, this is because what we're looking at is the multiple sequence alignment from an engineered phage, and it's been engineered to express or to have in its genome a sequence that is a unique molecular identifier that is different and random for each individual in this population. And in contrast, you see the other regions represent the conserved rest of the genome, which is the same among individuals. And actually, you can see here, there's another barcoded region, smaller region here. So you can see how you get some interesting information from these multiple sequence alignments. One thing you can get is consensus. And you can see a type of consensus that's represented as a logo down here. And this can be useful for example, for improving the accuracy. So in this case, that's happening. We have some various errors and things that you can see, some noise basically in the data. And the consensus here then is kind of clearing out some of that noise, and we're getting the most representative bases in this case. And multiple sequence alignments can also be used to assemble larger sequences from overlapping fragments. And it can be used to discover relationships between related sequences, common patterns, and domains. So first, let's talk about the consensus methods. And if the sequences are aligned already, then this is often acceptable for many applications. And the easiest version is what's called the dumb consensus. And basically, it means you just take the most prevalent base in each column as your final sequence. So for example, in this column, we have I and L, which are competing for representation in this column. But since there are more I's than L's, then the consensus sequence reads I in this particular position. And for many applications, this is fine. Dealing with deletions and insertions, you can do in different ways. So in this case here, we're taking the gaps added here as the majority winner of this column. And even though there's a gap in this sequence, we are taking E as the majority winner in this sequence. But there's an alternative to the dumb consensus, which is slightly more nuanced. And this is something called the pattern or prosite pattern. And this is a kind of generalization of the consensus model. And the idea is simply that whenever there is contention for a particular position, a disagreement, like a non-unanimous base, then we would try to represent that information in a condensed format. So we're still printing out basically a single linear sequence here, but it's got some more information. So for instance, in the third position here, we have both K and H bases. And X's here can be used to represent any base. So this would be a position where there was no obvious consensus. And we can use numbers here to indicate multiple bases. And so this would be like 2 bases of this here. And yeah, so here are the rules. You can see them. We can also disallow certain amino acids with these braces. And yes, so in general, this can be a nice way to represent with slightly more nuance a consensus, an alternative to the basic dumb consensus. And here you can see an example where we've attempted to construct one. It's a bit shifted or offset here, but we can see that this second to last column here, there's not much agreement as to what is going in there. So we've put an X. Likewise, we have this column here, which has F's and L's comprising the column. So we put both F and L in square brackets at that position. And then in the other positions, because there is agreement, we're just taking the unanimous base that is present in those columns. Frequency tables. So if we have a multiple sequence alignment, we can take, we can create a matrix where each column corresponds to a position in the multiple sequence alignment. And instead of the rows being the sequences, as in the multiple sequence alignment, we can condense the information into frequency tables. So we just list out our bases and count basically the occurrences of those bases at each position. So in this particular column, we saw, say, 61 A's, 145 C's, 152 G's, and 31 T's. And this can reveal patterns. So for example, the Tata box is this A and T rich sequence motif that is well studied and which frequently occurs about 30 base pairs upstream of RNA start sites. So here you can see promoter sequences up here. The Tata box that we're interested in is occurring here. And here we've got our exons and introns. And it might look something like this, where we have T is highly represented here, and then A is highly represented here. And then, yeah, so we see this kind of alternation pattern, and in general, a lot of T and A material. So this can be how a frequency table might reveal that information. And how do we know if it's significant? So it occurs in 78% of analyzed promoter sequences. A question might be, how do we know this is meaningful? And the answer is, like many of the scoring systems, to compare the frequencies with a background or the expected frequency. And so background frequencies have this Tata motif just present in 5%. So then we can see that 78% is quite significant. And you can express these frequencies as log odds scores. in something called a position weight matrix. So it is similar to a frequency table, but instead we're looking at a log odds score. And again, like with many of the scores, like the scoring system in Blossom and PAM, we're taking one frequency or probability, and we're taking its ratio with another frequency or probability. And in this case, it's, as always, it's our kind of frequency of interest divided by the background or expected frequency. And again, as with scores, we take the log to make for easy tabulation or easy arithmetic. And yes, so that's position weight matrices, a slight variation of frequency tables. And then we have another object, which is something called the position specific scoring matrix, or PSSM. And this is another kind of profile. And these are, you can think of these as an extension of substitution scoring matrices. So in these last examples, we're not thinking about substitution at all. We're just looking at observing the frequencies there. So to think about it, it's good to remind ourselves a bit about substitution scoring matrices. And in substitution scoring matrices, right, you have, you're looking at an alignment between two residues, A and B, and you have a fixed score for that pair, the score, the AB score, right, SAB. And you're not really considering the context or the environment that A and B are in. In alignments, you also have gap penalties that are kind of fixed. Maybe you have an opening and extension, but the position in the sequence doesn't influence any of the scores involved. So position-specific scoring matrices, on the other hand, take into account all of this spatial preference, like the position information matters for position-specific scoring matrices. And it's similar to a regular substitution scoring matrix, so you're still going to have substitution scores for each amino acid pair. But here we're thinking instead about the probability distribution for your profile of amino acids and how it changes at each alignment position. So we have this frequency that is specific to a column and to a base. So for each base, we would have a different frequency, and for each column, a different frequency. And you would just calculate it by counting the number of that type of base in that particular column and divide by the total number of sequences. And then you would compute a score for this matrix where, and the matrix would have this format of being such that each row is a particular residue type, particular amino acid, and each column represents a position, an alignment position, and then each matrix element corresponds to a sum of substitution scores for a residue substituted with every residue type B weighted by their respective frequencies. And it looks like this. So like you would sum for all bases of a particular type, you sum the frequencies in that column times the scores for that base with the base in each row that you're interested in, the base of that particular row. And then this gives you this M for a particular column and for a particular amino acid. So let's do like a quick example. So here we have this rectangle here is our multiple sequence alignment that we're kind of imagining here, where it's got multiple rows representing sequences and columns representing positions in those sequences. And then if we were to take a look at this particular column, we would see that there we can count the number of amino acids of particular base type B, and we count them up, and then we compute then for this column and that particular base type a frequency. So just divide by the total number of sequences. And then we can then use that. Oops. We can then use that frequency over here, and we multiply it with a substitution score, and this would just come from Blossom or PAM, etc. And this is for a particular pair. And so right now we're thinking about a particular base that's represented in our column, and then we're looking at that base and its substitution probability for a particular base in our position-specific score matrix. So our position-specific score matrix would have, rows here that correspond to each of the different types of amino acids, and then we would, for each element there, for each column, for each position, we would then compute one of these scores then for that particular amino acid based on the content that we saw in that particular column. And this gives us this context-specific substitution frequency. So here's an example of what one looks like. And again, with these scoring systems, what we can do is to get another log odds representation. So we're looking at orders of magnitude then instead of actual probabilities. And the way you read this is, if we go down here, it's like each of these columns then is an alignment position in the original multiple sequence alignment, and each of these rows here is an amino acid, and you can see how the values are changing. So it's not just one substitution score. It's really context specific. So here's an example, a mini example of how we would make one. So let's say we're looking at this column and it's all L's. So in this case, the frequency is 1. So to compute the values that would go into the position-specific score matrix, it's very easy. All we do is multiply 1 times the substitution score for this particular base, L, with each respective amino acid in the table. So substitution of L for A, and for L for C, and L for Y, and so forth. But then what happens if we don't have a unanimous column? And so here we have F and we have L. And in this case, we have to calculate the frequencies and basically weight the scores accordingly. So we have a score for F substituted for A and L substituted for A. But because there are more F's in this column, we weight it accordingly. So we're using the frequencies here, 4 out of 5 residues here versus 1 out of 5 for this one. And so that gives us a different value than we would have if it was just all F, for example. So you can find examples of these if you go to this pro site and you check for a particular entry. Here we can see the multiple sequence alignment, and you can see how some columns are more or less in agreement, some more than others. And ProSite then will generate a PSSM representation for that multiple sequence alignment. And so here you can see one of the actual results. So if you're wondering why this is useful, the insight that's offered by PSSMs is really from this context sensitivity. So you can think of it like a substitution matrix, but it's really taking into account the fact that substitution probabilities are changing according to the position, and that this context has some influence. And if you think about where that comes from, this is because the structure of a protein might influence the substitution probability. So it's not just the basic physical-chemical properties that influence amino acid substitution probabilities. Of course, there is that. So, negative residues are maybe more likely to be substituted for other negative residues, for example. But the context also has a huge influence on substitution probabilities as well. And this will take that information into account. Then we have something called the sequence logo. And you may have seen this before, and it's very intuitively easy to understand. So we can see from this that G is really prevalent in the particular position here, and that these very small ones we're less certain about the content there. And in some cases, so the height here is basically giving us some information about how certain we are. And the way this height is calculated is using information theory. So it has units of bits, and it's calculated using this information equation, which can be thought of as a measurement of uncertainty. So basically, the probability of getting a particular base tells us a bit about our uncertainty. So if we, for instance, if we're flipping a coin, we have, say, greater certainty of the result than rolling a die, because there are only 2 possibilities with flipping a coin, whereas there are 6 possibilities with rolling a die. Likewise, if we have uniform chance or uniform probability of A, T, G, or C, that is more uncertain than a situation where we are looking at mostly G. So like a situation where we're quite certain that we're looking at a G. So you can use this equation then to compute in bits the information content, and this determines the height. So for example, if you have a position that is 15% A and 85% T, then we have more certainty than we would about a position that is just 25%, 25%, 25%, 25%, right? We know it's probably going to be a T, so we'll be less surprised to see a T than we would be if it were uniform. And so you calculate it first with the uniform calculation in mind, so just filling in this equation here, P log P, for the uniform case, so just 25% log base 2 of 25%, and then times 4 for each base, and then that minus our observed information, so the 15% log base to 15% and the 85% log base to 85%. So and you compute this and you get 1.39 bits of height. So that would be, somewhere around here, like maybe similar to this T here. That is, we're fairly certain about the presence of a T. And if you recall from the beginning example, we had this logo down at the bottom, and so the same thing is going on here. So we have high randomness here. So the uncertainty in this case is very high, and the height then ends up being much lower in the logo. And then finally, we can talk about how these concepts get used in database searching. And there's this idea of how you can use a consensus or a pattern to perform a better search than you would with just a single sequence as your query. So imagine A is a particular gene sequence that we're searching for, and we do like a blast search, and we get a bunch of related homologous genes as our result. And it could be that if we had just searched for A, we might miss some other members of this family which are related to B, but not related enough to A to meet the threshold. So when we're searching with only one sequence that's part of this kind of network of related genes, we might not necessarily be representing best that family with our single sequence. Maybe we're looking at a kind of a peripheral case, a gene that is only distantly related to the core of the family. And so a solution to this is to do an iterated blast. And what you have is something called PSI-BLAST, or position-specific iterated blast. And the way it works is you first do a blast, a regular blast, and you use an e-value threshold or cutoff to select certain sequences. And then you iterate this process below. So you would iterate, you would make a multiple sequence alignment, generate a PSSM or a consensus from those sequences that you found, and then using that consensus or using that pattern or PSSM, you would then go back and search your database again, now that you have a more representative format or representation of that particular family that you're looking for. And then on the next round, you blast again with that, and you get maybe more hits that you missed in the first initial blast with just the one sequence. And then you repeat this process and you keep doing it, and eventually you converge until you've kind of exhausted all the members of this family. So yeah, just to summarize that. So you search for a pattern or a sequence of interest, use the pattern to search the database, you collect the results, do a multiple sequence alignment, define a new pattern based on the results, and then hopefully at the end you get a really nice consensus or PSSM pattern or logo for that particular family. Yes, so in the next video, I'll be talking about the approaches for obtaining multiple sequence alignments, as well as some topics from phylogeny. And the thing that kind of ties these two together is the use of trees and hierarchy. So I will see you in the next video with that.